what in the world has your life been like? You go through COVID and you're the point person for testing, I don't know, 2 or 3 million samples. And now you're just breathing. And then this hits you. And now you're the point person, I would imagine, or one of the point people on this thing.
It has been a roller coaster for sure. When I thought that things were, calming down and getting to a normal level of activity, HPAI hit, and here we are all over again. Not as bad as COVID, but we have been quite busy with it as well.
Hello, and welcome to the special episode of Dairy Digressions, a podcast of the American Dairy Science Association, a journal of dairy science, and JDS Communications. I'm Matt Lucy, former editor-in-chief of the Journal of Dairy Science and JDS Communications, and fellow of the American Dairy Science Association. I will be your host. Dairy Digressions is where we get an inside look at the science and the scientists that are making headlines from inside and outside the dairy industry. We explore the cutting edge research that creates knowledge and drives innovation in the production and processing of milk for human consumption. Mark your calendars. The 2025 ADSA annual meeting is happening June 22nd to 26th in Louisville, Kentucky. Abstract submissions are currently open from now until February 12th. I can confirm that there will be a special animal health symposium entitled Interspecies Crossover of Highly Pathogenic Avian Influenza H5N1 into Dairy Cattle. That's the topic of today's podcast. We also have a special issue on this topic now out in JDS Communications. Check it out at jdscommune.org. Today, it is a tremendous pleasure to have with us Dr. Diego Diel, who is an associate professor in the Department of Population Medicine and Diagnostic Sciences in the College of Veterinary Medicine at Cornell University. Welcome, Diego.
Thanks, Matt. Thanks for having me. It's a pleasure to be here to, talk a little bit about the work that we have been doing on this virus and, in my lab here at Cornell and also in collaboration with other groups in the US and abroad.
Yeah, it's a tremendous story. Diego, you're among friends. I'm a Cornell grad. I graduated in 1982 with a BS in animal science. So I'm familiar with Cornell. I know your pain in the middle of winter. Okay, I know these things. You have I noted from your CV, you've got a DVM as well as an MS and PhD in virology from the Federal University of Santa Maria, Rio Grande do Sul, Brazil. That's in the south of Brazil. You're south of Sao Paulo. It must be... I don't know if we call that quite tropical or what you would. Is it tropical or?
It's temperate climate. So we do have a mild winter in Rio Grande do Sul. It would be probably similar to a little colder maybe than in Georgia or Kentucky.
So I see that your research interests, you're basically a virologist, you're interested in virus-host interactions. Your goal is to understand these interactions at the basic level so you can improve vaccines and vaccine vectors for disease control. And of course, you're interested in diagnostic assay development and livestock feed biosecurity. What I think in a Google search I found out, how you did this stunned me. You were director of the Cornell COVID-19 testing laboratory, and you were testing 5,000 samples every day, 7 days a week during the pandemic. Wow.
Yeah, it was quite a daunting task. We ended up testing over a course of 3 years that the lab was functional and operational. We ended up testing over 2.1 million samples.
We're going to get to today's topic, but how did that work? Did the president of the university come to you, or how did that conversation go? I'm just curious.
Yeah, the current interim president of Cornell, Mike Kotlikov, he used to be affiliated. He was the provost at the time of the pandemic, and he was also the dean of the College of Veterinary Medicine. So he's a veterinarian, is affiliated with the CVM here at Cornell, and he was aware of the work and the diagnostic expertise that we have at the Animal Health Diagnostic Center. So he reached out to the dean of the college, Dr. Dean Warnick, And they came to HTC, to Francois Elvinger, who is the executive director of HTC, and asked if we could help setting up the testing program at Cornell. This was at the time that the university was closed, and they wanted in the spring of 2020. And the plan was to institute this testing program with a testing lab at Cornell that would then allow the university to reopen in the fall semester.
I think it's a tremendous story. I'm sure we could do a whole podcast on exactly how do you ramp up to that level of testing. And I think it kind of points to the importance that colleges of veterinary medicine can play in diagnostics, both animal and human. Cornell, the Cornell Med School, where people might think you would go for something like that, but maybe not, is in New York City. It's not in Ithaca. I just think that's a tremendous story. Congratulations. I was watching that video going, yep, that's how you do it. All the robotics that you guys developed. Must have been quite a moment.
It was quite a moment, but I'm really glad it's over now and that we're basically over that pandemic. It was a very unique experience. I mean, obviously, this was not a task that I performed alone, and there were a lot of people in the college and a lot of expertise that were brought together to make it happen and to be able to integrate diagnostic workflows that we use in the veterinary world to do human testing. So we also we partner with the local hospital here, the Cayuga medical system, and did quite a bit of testing for them as well throughout this period.
Quite a story. Today we're going to talk about, well, we're not talking about SARS. We'll talk a little bit about SARS-CoV later, but we're talking about these nasty viruses. They're just awful things. Today we're talking about H5N1 outbreak in dairy cattle. And of course, H5N1, you hear a lot of different names, highly pathogenic avian influenza virus or bird flu. It's all over the news. We're talking about the outbreak that began in Texas in early 2024. And the same virus is creating headaches in the state of California. Governor Newsom declaring, I think last month, a state of emergency. California veterinarian Dr. Annette Jones has issued a statewide ban on dairy cattle and poultry exhibitions. Let's remember, California's got 1.7 million dairy cows, and there's more cows in California than any other state. And I'm not sure if it's fair to say they've been hit the hardest, but they've been hit hard, Diego is a very humble individual, but basically his group, in collaboration with others, which you've already said, is publishing some of the really frontline information on this outbreak. I guess I'm not a virologist. I'm a cow guy, I'm a reproductive biologist. Okay, I did a lot of reading this past week preparing for this thing. But you guys are on the front line, and you've had some very high-profile papers in Nature, and you've been quoted in the New York Times, and all over the place. So, but I want to just kind of have you take us through. We're going to kind of shoot for somebody who's like me, maybe a dairy scientist whose expertise is not virology. And we're going to kind of go in that direction. So so what I did was I read a series of your papers and looked at them. I'm going to start with your journal, the paper you published in the JDS Communications. And this came out, a couple of months ago. And the lead author is Salman Butt, is he a postdoc in your lab, or who is Salman?
Yeah, Salman is a current postdoc in the lab. And he's one of the postdocs that was involved on the sequencing of the samples from some of the initial outbreaks in Texas on that first Nature paper that came out describing the outbreak.
Okay, so you have to remember, you're talking to a reproductive biology guy, okay? And I've done a lot of reading, but I was always confused. We're talking about influenza A virus, fair enough? I guess there's different kind of viruses, and influenza it's a flu-like virus, yes?
Yeah, so there are 4 types of influenza viruses: A, B, C, and D. Those are just different genera of the virus and all 4 types infect humans. Influenza A is the most prevalent one, and it's also widespread in different animal species.
So is this when I go get a flu shot, is it influenza A I'm dealing with?
Yes, but there's also flu B and C types in the vaccines that humans receive.
Okay. So we're talking about, they always have these letters, these H5N1, and I know you virologists, you put that in
Yeah.
there just to keep the rest of us confused. So I had to do a bunch of reading, okay? The
Yeah.
H stands for hemagglutinin, right?
Yes,
No,
that's correct.
and that's important. There's I guess 19 or 17, 19 variants of hemagglutinin.
Yeah.
And
Yeah.
that's important for the virus actually binding to the cell or something like that.
Yeah, so the hemagglutinin is basically the protein that interacts with the virus receptor in the cell and that, allows that initial interaction that will lead to entry of the virus into cells. If we can make an analogy, the HA of influenza would be the equivalent of the spike protein of SARS-CoV-2 that we
Okay.
all heard a lot about during the pandemic.
Okay, so there's this H5, and there's 19 different varieties, but H5 is one of them of this
Yes.
hemagglutinin molecule. It interacts with something called the sialic acid receptor. And this
Yes,
is
that's correct.
not, we're not going to talk about it yet, but this is important to the story here. This is very important to
Yes.
what's going on in cows versus humans, I guess, understanding the sialic acid receptor. Is that fair that that's important to this process?
Yes, it is because that's one of the main host determinants of the virus tropism. Basically, where this receptor, the sialic acid receptor, is expressed is what dictates where the virus is able to, which cells the virus is able to infect. Yeah, so it is very important.
So H5 is important. The
Yes.
sialic acid receptor is important. Now, this, for those who haven't had biology in a while, a lot of times we learn about different phages, and I just don't know anything about virology. They come in different shapes and stuff, but nonetheless, the basic process is this virus basically binds to the cell surface, injects I guess its DNA into the cell, uses other cellular machinery to make proteins and create virus particles, so you get more viruses. Is that fair, or what's going on here?
Yes, in the case of the influenza, would be the RNA. So the genome of the virus is made up of 8 segments of influenza A is made up of 8 segments of RNA.
Mm-hmm.
But that's basically it. After the interaction of the HA with the cellular receptor, the virus will actually fuse with the cell and injects its RNA genomic material into the cell. And that leads to, initiation of the replication of the virus.
So then you have this N1 stuff, this N1 that stands for neuraminidase, and that has something to do with the virus exiting the cell or getting out of the cell.
Yeah, so the neuraminidase is another very important protein in the influenza infection life cycle because it has an enzymatic activity that will actually cleave the sialic acid receptor as the virus is trying to exit the cell after completing the replication in the infected cell. So
So
that
you,
enables the virus basically to be released from the infected cell and infect other cells or be transmitted to other, susceptible animals or people.
so when we talk about this one, it's H5N1, and I know there's other clades after it, which we'll discuss in a second. But a couple things I just want to dissect away. When it does this, does it necessarily, well, in a bovine, let's stick with the bovine mammary, does it kill the cell? When it's done, is the host cell dead at that point?
Yes, the especially the highly pathogenic strains of the virus, they are able to basically lyse the cell after replication, and this is exactly what we see in the mammary gland. And in the mammary gland, the virus targets specifically the milk secreting cells, which
Yeah.
are the cells that express high levels of the sialic acid receptor. So the virus will attached to the cells, enters the cells, infect, replicates in those cells, and then it basically lyses the cells after the replication cycle is complete.
So this, when the reason in dairy, and we'll get to this in as we progress through this, the reason in dairy we see such a dramatic effect on milk production must be in part because these things are really doing a lot of I know you had a histology there. There's a
Yes.
lot of damage to. It's not like, oh, I don't know, there's a lot of damage to the mammary epithelial cells, which make the milk associated with this infection. Is that fair to say?
Yeah. That's correct. Yeah, there's complete destruction of those milk secreting epithelial cells, which leads to a very severe mastitis and destruction of that epithelium.
Yeah. And so it's not we'll get to this, but I'm just curious how cows recover from that. I mean, if we think about the mammary epithelial cell, we think about the lactation curve, these cells over time, part of the reason milk production goes down over time is because they lose cells. And now you have this virus stopped, which is completely, which is just super, super interesting to me. Before we move on to some other questions I have, and we're working our way through this, the swine, okay, H1N1 2009 pandemic in people, swine flu.
Yeah.
Does that have anything to do with what we're talking about today? Are there any parallels to that situation? Or are we just, that's a whole different ballgame and it doesn't apply?
So it's a similar situation in which we have observed a species jump. In that case, the virus actually jumped from swine to humans. It was not a highly pathogenic avian influenza virus. It was, was also an influenza A virus. That particular virus had genetic material or genome segments from avian, swine, and human. So it was a triple reassortant virus that was most likely generated in swine, which are considered a mixing vessel for avian and mammalian viruses. And after that reassortant virus was generated in swine, it jumped into humans.
So I have this on my notes here. You called it a mixing vessel, okay, for both human and avian viruses. And in swine, in my reading, swine seems to be the one. What is it about swine that makes it so good at combining these avian and human viruses together, for example?
So swine are unique in that they express receptors that are used, sialic acid receptors that are used by both avian and mammalian viruses.
Oh.
So there's two types of sialic acid. And basically the difference is where a sugar group will be linked in the molecule of the sialic acid. So there's the alpha-2,3 and alpha-2,6 sialic acid types, and swine express both of them. Alpha-2,3 is favored by or is preferred by avian viruses, and the alpha-2,6 is preferred by mammalian influenza viruses. So swine express both of them, and if they are exposed to an avian and a mammalian virus at the same time, those two viruses can infect the same cell. And because the genome is segmented as the viruses are replicating in the cell, chances are that you're going to get a virus that is a mixture of both viruses coming out of that infected cell. And then that could be a completely new virus that did not exist before.
That is the best explanation of a mixing event I've had all week. Okay, so you must teach virology or something, because I finally understand it. So they carry both the avian and the mammalian, I guess, or human, and they get into the same cell, and basically they're re-putting together their virions, and you get a little bit of a mix there, and that's a new virus.
Exactly.
Yeah. All right. So that's important to this story as well. H5N1 clade 2.3.4.4b. What is the clade all about? What the heck? So now for you reproductive biology people out here who have turned off the podcast already because it's too complicated, just kidding. Okay. I can say that because I'm a repro guy, but what the heck's the deal with the clade? What is the clade all about?
So the clade is basically just the genetic lineage that these viruses actually belong, and is based on the sequence, the nucleotide sequence of the HA protein or gene in this case. Different lineages will emerge as the virus, as influenza virus, replicate in different host species or within the same host species, as the virus replicates, it accumulates mutations over time. And, when they accumulate an X number of mutations, they may form a new lineage that is genetically distinct from from the lineage.
So that has to do with the family tree. So this is like,
Exactly. Exactly.
this is your clade. This is your heritage, your clade. So that's kind of like the family tree. And then of course, lucky us, this was over in Europe somewhere, I think, or I don't know where it originated, but maybe it was in Europe. And then you get some random gull who decides it wants to fly over to America, probably got blown over to America in some sort of storm in February 2022, lands in the Americas and spreads the clade to wild ducks. Is that correct? That's how it went down, a sort of an event. So this one gull, how do you know it was one gull? How do you know it wasn't 20 gulls?
It was probably not one gull. I think it was probably a group of birds that flew into North America. The first detection was in December, actually, in Canada. December of 2021 in Canada, the first detection of the clade 2344B in North America was in Canada, December of 2021 in Canada. And then January, I believe it was North Carolina, the first detection, again in wild birds, both of these events in wild birds. Soon after the detection in North Carolina, in wild birds, it was detected in domestic poultry, and we have been dealing with this particular clade since then.
Yeah, so this is the avian this clade kills a lot of poultry, and of course, as you stated, I think on this article, 100 million poultry deaths in 48 states. So this gull or group of gulls comes over. All of a sudden, it works its way into ducks and then poultry, and you got 100 million deaths. And in poultry, this particular H5N1 clade, it does not make a bird happy. Is it lethal, or are they die because they are depopped, or they die because they die from the virus?
I mean, it's a highly pathogenic virus for domestic poultry. Right now, I think we are at over 130 million domestic birds or poultry that have been affected by this virus. Most chickens and turkeys that will be infected will die very rapidly within 3 to 5 to 7 days post-infection. This large number of affected birds in the US, 133 million birds affected, not all of these birds were actually infected and died of the infection. But the control measure in poultry, it's different from what has been adopted in dairy cows, for example, is that once the virus is detected in a poultry farm, all the birds in that farm are cooled in order to prevent further spread of the virus. So there's a complete slaughter and clean out of that farm. It has to remain depopulated for a certain number of weeks or months, which I can't remember right now what's the procedure. But that's really a measure to prevent further spread of the virus once it's detected in a domestic poultry farm.
So it's my understanding that this clade then, we've got it in wild birds, we've got it in migrating birds. It's my understanding it sort of moved down the west coast of South America, moved back up the east coast of South America, killing wild populations of birds on the way, killing marine mammals along the way.
Yes.
This is all true, killing lots of different species, or it's okay, I understand birds, but why would it kill a marine mammal? I don't understand that. I mean, it's a seal or something.
Yeah, and it has caused quite significant outbreaks in marine mammals, exterminating certain populations in South America. The reason for that is unknown. I think there is opportunity for contact. These mammals have contact and they feed in birds as well. So I suspect that that's one of the ways potentially that the virus got introduced into these populations. And I think that is just a matter of the virus, finding a species that is susceptible to it and most likely expresses, the high levels of the receptor leading to mortality. In marine mammals, I don't remember exactly what's the clinical presentation that is observed in marine mammals, but in most of the terrestrial animals that are affected with this virus, a common feature is involvement of the central nervous system, which ultimately leads to the death of those animals that are affected.
So when we, I think some people might be aware of the situation with the cats on dairies. Are often they drink some milk from infected cows and it's lethal. So it is of interest, the fact that whereas this might kill a seal or a cat, it doesn't kill a cow. It has a totally different place. It infects the mammary gland, which is interesting, I guess.
Mm-hmm.
It's interesting.
I would say that mortality in dairy cows is probably underreported. We have monitored a few herds, and this is unpublished data, but we're currently working on a manuscript on this, where we did observe an increase in the risk of mortality of animals that are affected. And this is likely due to the severity of the mastitis that the virus induces in the affected cows. Mm-hmm. So in this study that we conducted, we monitored one farm that was affected, and the risk of death increased, to about 6.8 to 6.9% of what was normally observed in that particular herd.
So there was when one of these things jumps out of a certain species into another, we call it a spillover event. And the spillover event, I think, has been traced to Texas. It's a genotype. So now we've got a clade, but now you're mentioning a specific genotype here, B3-13. That's just a subtype of that clade, or a specific genotype of that clade. Is that how that works?
The genotype classification in influenza is based on all the 8 segments of the genome.
Okay.
So what's the constellation of those segments that make up that virus will determine what's the genotype of the virus. And then it's a very complex and complicated classification system because the virus has these multiple types of hemagglutinins that we talked about in our minidaises, and also has 8 segments of the genome. This genotype B313 emerged, I think, in October, November of 2023, and it had been detected only in a couple of wild birds in Wyoming, I can't remember now, and then California, and then in a skunk in New Mexico, and right after that, or around the same time, it was detected in the dairy cattle.
Do you think was it a single spillover event, or was it multiple spillover events in Texas, or just a single spillover event, or do we know that?
I don't think we have the data or the samples to determine that accurately. Unfortunately, the virus was circulating, causing this outbreak for about a month or a month and a half until it was detected. And samples from perhaps the first farms that broke with this clinical presentation were not available at the time that the virus was identified. So that really complicates defining exactly whether it was a single spillover or not. What is clear from the data that we have now is that it seems like after that event in Texas or New Mexico, in that area, we don't even know exactly where that happened first, but it was around that Panhandle region. It seems like from there the virus really spread to the other states, most likely through animal movement.
So do you think, I noticed in your paper that you talked about New Mexico, Idaho, Colorado, Kansas, Michigan, Ohio, and North Carolina, and I know there was some very clear evidence. Okay, cows moved from Texas to Michigan, for example. We know that's what happened. Was there always a way to trace cow movement to these outbreaks, or is there some possibility that maybe it was a separate bird spillover in these other states, or was it always, okay, there was a truck that went to North Carolina, dropped cows off? Do we know that?
In the initial cases, like in the initial interstate spread of the virus, there has been, documented evidence of animal movement to those states. In our manuscript, in the Nature manuscript, we documented that, where animals were moved from a farm in Texas to Ohio. 42 lactating cows were moved. They were moved before the outbreak was detected in the farm in Texas, and a couple of weeks before the outbreak was detected in Ohio. When we sequenced the virus from these two farms, we realized that it was the same virus, that they had very close genetic relationships.
There's a couple things I don't understand. How hasn't this gotten into Wisconsin and New York, for example? I do not understand that. Okay, you got all those cows. Wisconsin is packed with dairies. Seems like a perfect place for this virus to go. Upstate New York, dairy farms everywhere. How does and viruses? They are really good at getting around. How do they not get into Wisconsin? How does it not get into New York? That confuses me.
That's a very good question, and I don't think I have a good explanation for that. It could be the biosecurity measures that were implemented at the state level, requiring, testing of all the animals coming in, that were moved across the state. So I think that's one reason that contributed to preventing the introduction of the virus. The spillover from birds to cattle, it's probably not a simple event. It involves many factors. So it's not something that would happen over and over again. And I think so that's another factor that probably contribute to this outbreak not being spread out yet throughout the all the states in the country where dairy industry is present.
I think one of the things that we need our listeners to understand is it's one thing for the cow to give it to maybe a person. And we know that there's cow to people transmission causes conjunctivitis. Yes. But once it gets into people, it doesn't spread. I don't think we believe this variant. If you get it, I don't think I'm at risk of getting it from you unless I try real hard. Okay. And that's important. But what's really important is the fact that a cow can give it to another cow. That's super important. That's what makes it different, right? A cow can give it to another cow, and they're real good at giving it to another cow, apparently. I think that's fair to say.
It's fair, and I agree with you. I think they are very good at transmitting the virus to another cow. We have, again, studies ongoing where we determine through serology that up to 88% of the animals in affected farms are exposed and get infected with the virus. Not all of those animals will present the clinical disease. In around 20 to 30% of the animals usually get clinically affected. But we have, infection rates of upwards to, the 88 to 90% in some cases. So there's very efficient transmission within a dairy farm between cows. But I agree, in humans, the virus, thankfully, is not been shown to be transmissible yet.
So in a person, I think it's true, they don't give it to another person. They might be able to give it to their cat or to their poultry. Is that correct? If that person with conjunctivitis walked onto a poultry farm, could he introduce the virus into the poultry farm?
Yes, yeah, I think that's possible. And not just through shedding of that virus, but if that person, and I think one of the most important ways of introduction of this virus in poultry farms is through fomites, clothing or boots or shoes or equipment that can also carry the virus and act as a fomite to introduce the virus in poultry farm. And poultry, again, are particularly susceptible to this infection. So small amount of virus, is able to infect these birds and initiate that outbreak.
So I'm going to ask you a question. It's not unusual in America to have dairy farms close to feedlots. I don't think we detect this in feedlots. I assume that's because cows are not lactating, and therefore it'd be very unlikely this virus would spread around in the feedlot. Or what is the story with that? We don't find any of it in beef, do we? Do we see this in beef?
I'm not aware of any reports, and I'm not aware of, I'm sure people are looking into this.
Yeah.
I'm not aware of anyone finding any evidence of infection in beef cattle. So it is another interesting aspect of this whole situation. Why just dairy cattle and not beef cattle? It could be related to expression levels of the receptor or the way that the virus is transmitted. So that's another question that we still don't know. Is it really only through the mammary transmission route, or does the respiratory route plays a role on this? So that's another question that remains open regarding this virus.
You had a paper in Nature where you kind of went through the evolution of the virus, a lot of genotyping, and kind of traced where it came from, and that all worked, and speculated on the process through which this virus came out of ducks, went into an intermediary host, which infected a whole nother group of animals, which eventually went to maybe another intermediary host that we don't understand, and ended up in the cows. And one of the questions I have, and we're going to move probably, we're going to talk about the other Nature paper where you did the infections, which is fascinating, where you actually took cows and infected them with the virus, and calves. I tell you, that paper, you learn a lot with that paper. I'm telling you, that paper is a good paper. All right. Yeah. And it's very relevant to the U.S. dairy industry, that paper. Mm-hmm. But I want to ask you this. I was in California, and there were some discussions about this. And one of the things that was said was, like, California dairies aren't just spread uniformly across the state. There's a bunch of dairies here, a bunch of dairies over there, a bunch of dairies over there. And the comment that was made was, if you have 10 dairies in a close proximity to one another, once it gets into that region, all the dairies are going to get it. And what I wanted to ask you was, and we don't even know for sure how it's transferred cow to cow. The assumption it has something to do with the milking parlor, or something that's happened in the milking parlors. There's so much virus in the milk that it's got to be a milking parlor thing. But how does it get from farm to farm? If a farm is 10 miles from another farm in California, how is it getting from? Is it the milk truck? I don't want to blame the milk truck, because the milk truck drivers will be after me, but how does it getting from a farm that's 10 miles apart? I mean, is it the birds? Is it what?
I think it could be all of the above. I believe that birds could carry it. I do also believe that trucks, may play a big role in the local transmission of the virus. And, I've always worked with the swine industry and with swine viruses. And if we remember the PEDV outbreak that happened here in the US in 2013, there are several studies showing and documenting that the virus actually moved with the trucks. And I think that in this case, we don't have those studies out yet, but I do think that if people would look into that and sample those trucks, they would probably detect the virus. And I think it may be playing a major role in this local regional spread of the virus that leads to the rapid, dissemination of the virus through those farms that are in that same region.
And then once it gets on the farm, for sure, it goes through the farm, as we've talked about, really quickly. Yeah. And as a virologist, even your experience as a virologist, you would agree those titers coming out of the milk of an infected cow are high.
They're super high. Yes. It's something I almost couldn't believe the data when we got the first results from the titrations. Some animals shed, up to 8.5 logs of virus per milliliter of milk. So you can imagine how much virus a single cow produces, a single affected cow produces, and releases in the environment once it's milked. So it's a huge amount of virus that each affected animal is producing. With that amount of virus, it's nearly impossible to prevent contamination of equipment or trucks or clothing of farm personnel that are handling these animals.
It's sort of maybe it's not fair to say unprecedented, but it's a lot of virus. I mean, far more
Yes.
than with COVID, even more than a COVID person might spread around or something like that. It's a lot.
Yes, much more than what a person infected with COVID would shed. Yes, for sure.
Yeah. I want to talk about your an additional paper you had in Nature. It was on the experimentally infected calves and cows, because I thought it was an important experiment that you guys did. And before I do that, I want to ask you a question. How does it work with Nature? Are they coming to you and your group and saying, "Hey, do you have anything new we could publish here?" Because Nature seems to really like this topic. How does that work? Does Nature go to you, or do you go to Nature?
Yeah, we went to them, initially with an abstract of the manuscript as a pre-submission inquiry telling the story, and when they received the paper, the abstract, the handling editor was very excited with the topic at the time, and invited us to submit the manuscript. And in fact, they expedited the review process of the manuscript and the entire handling of the manuscript. So we were lucky to be one of the first groups to detect this outbreak, and I think the completeness of this study that we performed also helped for the study to be accepted and published in Nature.
Yeah, both of these papers, and we'll have them in the show notes, were really nice papers, a really good paper. I really enjoyed reading them. And one of the things I really enjoyed was your strategy in the second paper where you're experimentally infected, you infected calves with nasal infection of the virus, assuming it was maybe coming in via nasal route. Those darn calves, you barely could move the needle with a big dose of virus in a calf. True or false? Hard to move the needle.
It's true. I just want to, just make a point here that this study was conducted in Kansas State through Dr. Jürgen Rick's group, and the dairy cow experiment was conducted at FLI in Germany by Dr. Martin Wehr's group, because they have the capability of running those studies in with large animals and BSL-3 facilities. So it was a very good collaboration, and I think that the idea of the study was to really try to document or answer a few of the questions that we initially had, and how the virus could the virus actually infect through the nasal route, or was it the mammary gland required for that? I don't think this study answers that completely, but I think it gives some hints into those possibilities. It seems like the mammary gland introduction or entry leads to severe disease and replication of the virus. Obviously, studies with adult cows, maybe dry milk lactating cows and dry cows, are still needed to assess some of these questions in terms of entry route and transmission of the virus.
I thought it was interesting in the calves, even though you did sort of a respiratory inoculation, there was some small responses, but sentinel calves, and I'm assuming those sentinels were in the same room, didn't
yes
get
they were.
the virus from their neighbors.
There was very limited replication of the virus in those animals, in those inoculated animals. The animals, the calves that were inoculated intranasally. So it's kind of a puzzle. We don't know for sure. Some field data, again, unpublished data that we have from a few affected farms, suggests that dry cows, so non-lactating animals that are present in these farms, during the outbreak, they also get infected. This is based on serological evidence. So obviously, these animals, in my mind, they can be getting infected through the mammary route. They are most likely being exposed through the respiratory or oral route, and getting infected that way. So the route of infection, I think it's a big question that also is linked to the transmission of the virus, and I think it's one of the biggest questions that remains unanswered on this particular scenario.
So when you inoculated the cows, for example, in that experiment, a large effect just basically recapitulated the disease, the pathology with the mastitis. And I thought the other thing that was kind of interesting, you had a couple of different inoculants. You used the current one that's circulating in America, and then you used another one that was from Europe or something, an older version, and it basically did exactly the same thing. Do I have that correct?
Yes, I think that was one of the most interesting findings of this study is that it's not just the genotype B313 that causes this severe mastitis. Other H5N1 viruses of different genotypes are also able to infect and cause the same clinical presentation, with severe decrease in milk production and severe mastitis in affected animals.
It's a great paper, and it's really easy to read. It's really informative, and we'll put that paper in the show notes. Hey, Diego, can you hang on for another 15 minutes? Do you have time to do that?
Yeah, sure,
I'd like
sure.
just to kind of talk a little bit about my last section of this, and that has to do with pandemic potential of virus. I just want to go through a few things. So bovine H5N1 isolated from the human eye is lethal in certain animal models, including ferret apparently is the model for these things. And so there are species that are very, very sensitive to this virus. It doesn't really seem to multiply. It doesn't seem like a person can infect another person. And I think it does seem like, I think most of the conclusions are that in people that do get infected with it, they can't really spread it, but the symptoms are fairly mild. Is that fair to say? The symptoms are mild for people that contract this disease?
Yeah, I think in most cases, I think so far there has been 67 reported human infections in North America, and there's two cases, one in Canada and one I think in Louisiana, where people actually got severe clinical presentations. The person in Louisiana actually ended up dying from the infection. The particular features of these cases, the case I think in Canada was a child, and the case in Louisiana that died from the infection was an elderly person that had other underlying conditions. So those are features that probably contributed to the severity of the disease. Thankfully, most of the cases have been, mild, respiratory or infections or leading to conjunctivitis. So it seems to be thus far the most common clinical presentation of this virus in humans.
And again, to restate it, if we talk about the multiplicity of infection, which is whether or not one person to give it to another, it's zero. It's basically zero. You're not going to pass it along. The other thing I heard, I'm kind of referencing a webinar I listened to earlier in the week from the Infectious Disease Society of America with Shira Dorona and Julio Figueroa. And they just did a webinar, and those are a couple of MDs talking about bird flu in the United States and some of these issues that we're going to talk about today. They mentioned, and even at this point, we probably have fairly effective vaccines for this virus in people. Like, are there some of the flu vaccines that we already have in production or available? Would a flu vaccine potentially protect us against this specific virus?
No, the seasonal flu vaccines wouldn't, because they are not, so they don't match the H5 subtype. Usually for influenza, you need subtype specific vaccine to elicit, robust protection. But there are probably stockpiles of vaccines that are based on the H5 hemagglutinin that would provide protection. The other important thing to remember is that we do have antivirals that are effective against influenza. And, obviously they were developed against the seasonal influenza viruses, but they have been shown to be effective against the H5 subtype as well. So this is different, for example, from the COVID-19 pandemic, where we didn't have any tools in the toolbox to fight the virus in the early stages of the pandemic, and in fact, for several months into the pandemic. In this case, I think if there was, to be evidence of transmission in humans, we would be able, the health, human health authorities would be able to deploy some of those tools to try to control that, spread of the virus.
So it's important to recognize that unlike COVID, where we were fairly flat-footed on that thing, we're not as flat-footed here with this one. And I hope I didn't misquote. I think you explained what they were probably trying to explain to me in this podcast. And and that's another thing I've heard, is that people have to understand we have antivirals in place that probably work pretty well against this thing. That doesn't mean we shouldn't take it seriously. So the way I understand it, and I'm looking at a recent publication in Science. It was just a Science news thing from December 13th. And I hope our listeners understand that some of the major scientific magazines in America are really covering this thing. They're interested in it, and it's a very interesting topic. And the title of this one, we'll put this in the show notes: why hasn't the bird flu pandemic started? And they're talking about H5N1 and dairy. I mean, why hasn't it started? And they bring up a couple of things that have to happen in order for this to really hop over to people and become a pandemic. They talk about necessary mutations in the hemagglutinin, necessary mutations in the neuraminidase, and also necessary mutations in the polymerase complex. I mean, this thing has to change. Is it possible for it to change? Is that a possible thing, or is that just, look, it ain't gonna, there's too much that's got to happen here? Or what are your thoughts on that?
Yeah, no, I think that's right. This virus doesn't have yet those critical amino acids that make it transmissible in humans and more infective in humans as well. If we think about the number of farms that have been affected and the number of human cases reported so far, is a relatively small number of cases. So that also suggests that the virus is not very infective to humans. And then the reason for that is it doesn't have those critical amino acids that enables it to infect and replicate efficiently in humans. And also to transmit efficiently in humans, it would have to acquire those mutations. Can it happen? Can the virus acquire those mutations? I think yes. And I think the risk of that happening is higher now that the virus is replicating efficiently and transmitting efficiently in a mammalian host, and in a mammalian host that where humans, or with which humans have close interactions. So I think that's always, when that happens, the risk of the virus spilling over and acquiring those mutations is higher than if the virus had remained endemic and replicating only in avian species.
I was doing some reading, and as you know, the SARS-CoV-2 COVID virus is always in the news, and there was some recent sequencing in the Wuhan market, and that basically the epicenter is the Wuhan market, as far as everybody can tell. And I don't know if it was a podcast I was listening to, but they were bringing up the point that, look, there was a lot of people going through the Wuhan market. And so a lot of people going through the Wuhan market with species that were probably harboring this COVID virus. And, I think there's 1 million people in Wuhan, or 11 million, excuse me. And that's the other thing I think about to the dairy industry. A lot of these are very big farms, but very few, relatively few. You don't have 1 million people going by or anything like that, relatively few people. And so maybe that's helping us too. There's a relatively small population of individuals interacting with these cows. But the situation with SARS-CoV-2 is, I know it's a different virus. I know it's different, but it was the same sort of concept where you have these animals that are infected, and all of a sudden, boom, it hops over to people, and off you go, And I know you know a ton about SARS-CoV-2. So one of the things in this paper that you guys concluded with was, some important points moving forward. And one of them is we want to, and I just would like you to comment on this, is our best defense here is probably to figure out how to stop its spread in cattle. As a human population, is that our first thing, is to try to figure out how to stop it spreading in cattle? Or is that just give it up, it's going to spread in cattle type of thing? Is there anything we could do to stop the spread of this virus in cattle? I mean, it's working its way through California. Is it going to go to Oregon? Washington, Idaho? Is it just a matter of time before it's in New York, Wisconsin, or do we know that?
I think I agree with that statement. I think our best interest would be to stop it spreading in cattle, because if the virus continues to spread in cattle in a mammalian host and transmitting efficiently in a mammalian host, there is a higher risk that the virus will acquire those mutations that are required for efficient infectivity and transmission in humans. So I think that's why we should do whatever we can to prevent that spread in cattle. Can we do it at this point without vaccines? I think and even with vaccines, I don't think we would be able to develop vaccines that would induce sterilizing immunity. So the virus would probably still circulate, but maybe would decrease the level of circulation and, infections in dairy cattle. It's a challenging question to answer. Can we do it right now? At this point, I don't know if we can. I think there has been a few instances of infection. I think Texas reported an infection in the last 30 days again. So the virus seems to have gone back to Texas, or maybe never left Texas. It was still circulating there. The other factor that is important, I think, is that we don't really know what's the prevalence of infection. There hasn't been a lot of testing done other than in farms that are clinically affected. And I suspect that many farms that are clinically affected didn't test all the animals. So that's one of the issues. I don't think we understand what the magnitude of the problem is, or we don't completely understand what the magnitude of the problem is. And without that information, I think is challenging to establish effective control measures for the virus.
I think that's one of the things that sort of came out in all my studying for this podcast, was that there's a lot we do not understand.
Yeah.
I mean, even simple stuff like, okay, we think it's getting transmitted in the dairy parlor, but how does that work? Or how do you slow that transmission? And then we know it's got to be traveling from farm to farm. We don't know if it's birds, trucks, house flies. There's probably certain things you can say it's probably not, but still we don't. There's so much work to be done. Hey, before I let you go, is there anything else you'd like to add in terms of this thing? You've been a very good interviewee. You've told me that you've answered every question. You haven't dodged a single question. I mean, you're just like saying it like it is. So anything else you'd like to add, or something you think that someone listening to this podcast should really understand before we kind of wrap it up here?
I think one thing is that people should remain vigilant to this virus. And I would highly recommend producers that suspect that the virus could be present in their herd, that they should collect samples and submit to diagnostic labs to test. That would be one of the ways of, early detection and movement restrictions is one of the ways of preventing further spread and further economic losses to the industry. I do think that there aren't any studies out there yet, but the economic implications of an outbreak in a farm are not small either. I think there is significant cost to producers from farms that are affected with this. So preventing introduction is probably of the best interest to producers and to the industry.
I may have said it earlier in a podcast. I think I did, but just California was down 9.2% in milk production. Yeah. Those are big numbers, historic numbers. Okay, this is not a 1% change in milk production. This is a lot. Yeah. And I probably said that at the beginning of the podcast. I have just a kind of a couple of quick questions for you. So, what in the world has your life been like? You go through COVID and you're the point person for testing, 2 or 3 million samples. And now you're just breathing. And then this hits you. And now you're the point person, I would imagine, or one of the point people on this thing.
It has been a roller coaster for sure. When I thought that things were, calming down and getting to a normal level of activity, HPAI hit, and here we are all over again. Not as bad as COVID, but we have been quite busy with it as well.
I think what I would say we get a lot of young people listening to this podcast, and I think this is, if you're a young person and you don't know what direction you want to take your career or what direction you want to take your world, microbiology and the way these, not only bacteria, viruses, the way they work and how they function and the things they do, it is absolutely fascinating. It is fascinating. And it is so important. I mean, I think, Diego, you can make that point. I know you're always busy, but you can go from a quiet laboratory where you're studying your virus to all of a sudden the world is at your doorstep.
It is, I agree. I think, I have never thought, when I was in vet school, I never thought about being a virologist or studying virology. I always wanted to be a clinician and take care of animals in the field. But when one of my professors invited me to do a master's in his virology lab, I decided to take the risk, and I've been doing research since then, and I don't regret I've never regretted the decision that I made, because it has always kept me busy and also learning. So I've been learning a lot in my profession for the last 20 years, and I think that's very rewarding. So anyone that is thinking about or considering to do work with infectious diseases or microbiology, I would highly encourage them to talk to someone that they know, or that even if they don't know, they should reach out to people and discuss what their career goals are, because it is fascinating for sure.
Well, I really, Diego, I know you're busy, and I know it's getting to be dinner hour, and I see some little children running around in the background maybe, and I know they're like, Dad, we got to eat dinner now, okay? And I just want to say thank you so much. This has been a tremendous conversation. I think it's really gone well. I just appreciate your honesty and openness on this. And I'll be thinking of you, man. Your life is going to be a little crazy for a little while longer, I think. Okay. It's one outbreak at a time. How about that? We'll take one at a time. And we'll hope this one stays in cattle and gets resolved. So, but thank you very much.
Well, thank you, Matt. Thank you for the invitation. It has been a pleasure to talk to you about this topic, and I'm happy to jump in the future if you want me to join again. It was a pleasant conversation. I really enjoyed it too.
Thank you very much, Diego. All right, everybody, that brings us to the end of our show. If you enjoyed Dairy Digressions, be sure to like, subscribe, and rate us. And spread the word to a friend or colleague. If you had feedback, please let us know at adsa.adso.org. For show notes and more episodes of Dairy Digressions, and to learn about everything going on at ADSA, go to adsa.org. Until next time, until next episode, thank you very much for listening. Thanks, Diego.
Thank you.